So the next thing we're going to talk about very briefly is the pentose phosphate pathway, PPP. You may ask yourself, why? Well, the reason why we're doing it is because of the MCAT. It's on the MCAT. But the reason why we do it biologically is to get ribose and something else. Ribose, you may remember, is a pentose. It's a 5-carbon sugar. We're going to go from a 6-carbon glucose to a 5-carbon ribose. There's a decarboxylation event somewhere. Going to yield out CO2. Ribose, what's it used for? Nucleotide synthesis. Therefore, you're going to get precursors, building blocks, for DNA. All right, how convenient. It's also a sugar, but you get the idea. In addition to that, we get something else out of this. NADPH. What is NADPH? It's a reducing agent, somewhat powerful actually. So it functions as an antioxidant. We'll talk about the redox pairs in this in a second. And it can also carry energy if you needed it to. So how's it work? You know, in one situation, you're going to shunt glucose 6-phosphate out of the glycolysis pathway into making glycogen. You're also going to shunt a different version, or the same version of stuff with a different enzyme. So here, you're going to have glucose 6-phosphate dehydrogenase. Okay? And when you think dehydrogenase, you're going to think yielding NADPH in this concept. You're going to end up with 6-phosphogluconolactone. You do not have to have that memorized. It's just helpful to know. Okay, so 6-phosphogluconolactone, but because of dehydrogenase, you're going to also get NADPH. And there's a couple other steps. You may see these words. Don't have to have these memorized. Don't have to have all the other enzymes down either. So you're going to have a 6-phosphogluconate. There's another important step happening right here. Now this one is a rate limiting step, this first step here. Basically that means it's irreversible. The last one, not quite so irreversible, but still committed. Now like I said, we're going to get ribose out of this, but we're not quite there yet. So the step after this is ribulose 5-phosphate. How do we get there? Well, there's an enzyme. 6-phosphogluconate dehydrogenase. 6-phosphogluconate dehydrogenase. So if you have that, you're going to yield, again, NADPH. So you have two situations, two spots we get NADPH out of this pentose phosphate pathway. And then, lastly, from the ribulose, we get ribose 5-phosphate. Now, think about DNA and RNA. Ribose 5-phosphate. As written, these redox pairs. So this is reduced, which means this step is oxidizing. So this is the oxidized form, and that's reduced. Reduced, oxidized. Okay? So this is the oxidized form. So you may help yourself think about oxyribose 5-phosphate. Which means a different step entirely would give you the DNA part of deoxyribo. Get the idea? So this pathway is ultimately oxidative. It's anabolic. And it is effectively a decarboxylation. So if we're going to have a decarboxylation event, it's going to be yanking off a CO2. It's going to yank off a CO2 when we go from 6 to 5. So we have ribulose 5-phosphate, then NADPH, and this is where the missing CO2 was from. So that's the pentose phosphate pathway, nitty-gritty MCAT style.